Okay.
Perfect. Thank you for doing that.
No problem.
All right. So guys, let's go ahead and get started. So we are the main focus of the lab today is cellular respiration, but in reality you cannot talk about cellular respiration without even mentioning fermentation, because they're basically the two main modes for cells to get energy to make ATP. So that's why both are being mentioned. So I just want to show you guys that, because I think last time I did say that I was going to show you the distribution of the first quiz. And then also a reminder that we do have the next quiz is coming up next Saturday. So even though quite a few people did run out of time, the distribution is actually not bad at all. So, it's a 20-point quiz that converted into a 100 scale, because that's easier for everybody to understand. So there are 13 people who are above 90, and then 7 people that are in the 80 range. So that's half of the class, more than half of the class. 6 people in the 70 range, 2 people in the 60 range, and 3 people end up getting the score below 60. So that's kind of the distribution of the last quiz. So normally, in exams, people expect kind of a bell curve. So actually, this particular quiz, the score is actually higher because we have a lot more. We have more people in the 90 category than in any of the other category. Okay, so let's go ahead and talk about metabolism. So this lab and also the next lab where we're going to talk about photosynthesis is all about how cells get energy. So if you think about it from an ecosystem point of view, most of the energy in the ecosystem actually come from the sun. So basically plants, which are, and also algae, they are the producers, and they basically use photosynthesis to convert the light energy into chemical energy. And then, consumers like animals and fungi, we eat plants, or fungi are decomposers, so they eat the dead body of the plants, and then we can extract energy from the carbohydrates or the sugars from the producers. So the word metabolism basically describes all the chemical reaction that's happening in the cell. So it's just, at any given moment, there's a lot of chemical reactions that are happening. So that's basically metabolism. And then if you look at the chemical reaction for photosynthesis, so photosynthesis, basically the producers or the autotrophs use CO2 and water as the substrate, and then they produce glucose, and then produce oxygen also kind of as a side product. And cellular respiration, which is what we're focusing on today, which, by the way, animal, plants, fungi, bacteria, we all do cellular respiration, some form or another, and it's actually using glucose and oxygen as the substrate, and then you end up producing CO2 and water. So these two processes, photosynthesis and cellular respiration, are basically the converse of each other, right? So that's the substrate for photosynthesis, and they are the product of cellular respiration. And then these are the product of photosynthesis, and they are the substrate for cellular respiration. And then, of course, the main point of cellular respiration is to extract some energy in the form of ATP, so that the cells can use ATP to fuel different reactions, and also transport and stuff. Okay. Um, that's not bad. First Kahoot question of the day, and we've got 31 people giving the correct answer. So of course, you cannot talk about chemical reactions without talking about enzymes. And the question is basically, what do they do? And the enzymes, basically, they speed up chemical reactions. And I love Amoeba Sisters, so they make a lot of videos. And this is just one of their gifts to show you that the enzymes bind to substrate, and that binding is actually very, very specific. So if you look at the chemical reactions that are happening in the cell, they are not happening in isolation, right? So usually a lot of the chemical reactions are organized into these pathways. So, in this example, so compound A, goes through the first chemical reaction. It's being catalyzed by the first enzyme and then gets converted into B. And then B can actually participate in the second reaction, which is catalyzed by the second enzyme, and then gets converted into C, and then C, gets converted into D, and so on and so forth. So these are called metabolic pathways, and in the cell, most of these reactions are organized in some kind of pathways, and then different pathways will actually interact with each other, so they kind of feed into each other and stuff. And in general, catabolic, metabolism is basically divided into catabolism and anabolism. So catabolism is literally just breaking down large molecules into smaller ones. And so if you look at it, so for example, glycogen, it's a large polysaccharide, and then we break it into individual glucose molecules. So that's a catabolic reaction. And most of the catabolic reactions are actually energy releasing. So they're energetically favorable, and then there's some energy being released in that process. Anabolic pathway are the converse of those. So basically synthesizing a large molecule from the smaller building block. And most of these are actually energy requiring. So you have to put in energy for these chemical reaction to be able to happen. And when you talk about energy in chemical reactions, so we are talking about free energy, and so these are represented by delta G, if you guys remember from your, I guess, physics or chemistry. And in terms of the reaction in terms of the energy for chemical reaction, there are two different kinds. So in this one, the reactants actually have more energy than the product. So basically during the chemical reaction, some energy actually gets released, and these are called exergonic reaction. So basically, exergonic means that energy is released. Another term that's kind of similar is called exothermic. And exothermic basically means that energy can be released in the form of heat. So there are some textbooks that actually will use exergonic and exothermic kind of interchangeably. And then if you look at the bottom reaction, the reactant has lower energy state than the product. So basically for the chemical reaction to happen, you have to put in energy for this to happen. And these are called endogenic, basically energy requiring reaction. And you can see that, so the exergonic, because you can release energy, it could happen spontaneously. But in reality, a lot of times just because it's possible to be spontaneous does not necessarily mean that it is spontaneous. But the endogenic definitely cannot be spontaneous because you literally have to put in energy for that to happen. And so the reason that even if it is an exergonic reaction, it may not happen spontaneously is because, pretty much all chemical reaction, you actually require an activation energy to get the reaction started. So, if you guys look at the chart over here, So just to go from the reactant to where the chemical reaction can proceed, you have to put in that much energy. So that's called the activation energy. And this is really where enzyme comes in, because enzyme work to speed up chemical reaction because they can literally lower the activation energy required for the chemical reaction, and then basically can speed up the reaction. So this is actually one of my favorite analogy. So we're actually, talking about the activation energy. So think about the activation energy as a barrier. So you have these frogs, and they're amphibians, so they all want to get to the other side to go to the pond because they love water. But if the barrier is very high, only the most athletic ones can actually jump through that hoop and then get to the pond. But if you lower the barrier, so remember we're lowering the activation energy, most of the frog can actually jump through that barrier and then get to the other side. So by basically lowering that barrier, enzymes allow chemical reaction to happen faster. And enzymes bind substrate. So basically most enzymes are proteins, and the place where substrate bind is called the active site. And the active site, the shape of the active site and the shape of the substrate has to be a pretty good match. So that's why people sometimes call it lock and key model, because you really have to have very specific shape of the active site so that they can bind to each other. It doesn't have to be exact match, and that's because once the substrate bind to the enzyme active site, they actually both go through a little bit of conformation change, and then that allow them to bind each other even better. So that's called the induced fit model. So basically the substrate, it's a pretty good match with the active site, and then once it binds, it can actually induce even more conformation change so that they're even better fit, basically. So they form this enzyme substrate complex, and then the chemical reaction happens. So you end up having the enzyme product complex. And then the last step, of course, is the enzyme will actually release the product. The enzyme itself does not get consumed during this chemical reaction. So it can basically go back and bind to the next substrate and then catalyze the next chemical reaction. So enzymes are, being recycled all the time. Okay. So the question says, synthesizing glycogen. So remember, glycogens are made of many, many glucose molecules, forming a chemical bond. And so that is an anabolic reaction because you're going from single monomer into a large polymer. And it's also an inorganic reaction because synthesizing that large polymer actually require energy input. And so who do we have? We have Bahar, Charles, Roop, Malika, and Nina on the leaderboard.
Do you mind if I ask a question before we continue?
Go ahead.
You mentioned that for the substrate-linked enzymes that there is a little bit of structural change, but then you also mentioned that enzymes aren't consumed in these processes. So how is it that things can change a little bit, but they're not like actively taken out of circulation? I guess making those two things make sense at the same time is confusing to me.
No, because when the enzyme is binding to the substrate, it's catalyzing that chemical reaction. So while it's occupied with one substrate, it's catalyzing that particular round of chemical reaction. But as soon as it's finished and it's released the product, it can go back and bind to the next substrate.
Got it, and go back to normal. Great.
Yeah,
Thank
so
you.
being consumed actually means that it gets converted into something else. So it doesn't get converted into anything. It's just, the conformation change is also reversible. So once it releases the product, it goes back to its original conformation.
Got it. Thank you.
Okay.
Yep, thanks.
All right. So that's enzyme. So they basically, catalyze chemical reaction or make them happen faster. In a lot of the metabolic pathways, the chemical reactions belong to this category of reaction called redox reaction. So redox is basically an abbreviation of reduction and oxidation. So redox reaction happen in pairs. So basically electrons are being transferred. If you look at this graph, one of the reactant is the electron donor, and it donates the electron to the exact electron acceptor. So loss of electron is called oxidation. So basically, this guy is being oxidized, and then that's the product of the oxidized donor. And then gain of electron is called reduction, and then so this guy is being reduced because it gains electron and it gets transformed into this other form. On paper over here, it looks fairly straightforward, but people get reduction and oxidation mixed up all the time. So this is where sometimes these things like oil rig is very helpful. So oil stands for oxidation is loss. So basically, if a party end up losing electron, it's being oxidized. And then rig is, reduction is gain. So if a party is gaining electron, it is being reduced. And, if that's not confusing enough, this guy is being oxidized, and so this is oxidation, and that means the guy who is gaining, the electron is also called the oxidizing agent. And then the guy who is being oxidized is also called the reducing agent because it's donating the electron and then allow the other guy to be reduced. So there are a lot of termination over here. You basically need to figure out, where does the electron go? So who's losing electron, who's gaining electron, and then, which name kind of matches with their description. So if you think about how we get energy from molecules like glucose, for example, it basically is through a whole series of reactions that oxidize glucose. So if you take glucose, if you take sugar, and then you just light a match and then burn it, the sugar will burn, and then eventually it will basically release CO2 and water, and then a whole bunch of energy will be released as heat. What the cell does is, instead of just burning sugar in that one gigantic step and then releasing all the energy as the form of heat, it breaks it down into smaller steps. And then in these smaller steps, we can actually harvest some of the cellular energy and then use those harvests, mostly in the form of ATP, to fuel other reactions that are happening in the cell. So you can really think about the cellular respiration as kind of a whole series of reactions, gradually oxidizing sugar and then basically generating some ATP in the process.
I have a quick question, if you
Sure.
know, just about like the basic definition of cellular respiration. Is it specific to organic molecules only?
What do you mean?
Like only like inorganic molecules can also go through cellular respiration? Or no?
No, I think cellular respiration in general, we are mainly talking about glucose.
Okay, so, okay, yeah.
Yeah, we are mainly talking about glucose. I mean, the metabolic pathways, so in cells, if you read the whole chapter,
Yeah.
you know, it's more complex. So other things like amino acid can also give you some energy. But, you know, they kind of get converted into something similar to glucose and then feed into a similar pathway. So when we talk about cellular restoration, we are talking about glucose. Okay. Yeah. All right. So ATP, you know, it is the cellular money, the energy currency in the cell. And the structure of ATP, it's basically, it's a nucleotide, right? So you have a ribose over here. And so that's the sugar that's in RNA. And then you have the adenine, so that's the nitrogenous base. That's really similar to the A in RNA also. But, you know, instead of having one phosphate, it has three phosphate. And the energy is basically stored between the phosphate group, you know, because they're all negatively charged. So these covalent bonds end up being very high energy. And in cells, you know, when you break one of these covalent bonds, you release a lot of energy, and then you can use that energy over here, you can use that energy to drive chemical reactions, or also to drive like transport and stuff. And then you can use energy from catabolic reaction to synthesize new molecule of ATP, basically using ADP and phosphate as a substrate to synthesize new molecule of ATP. And in addition to ATP, which is, you know, cellular cache, there are also these other electron carriers, or, you know, also called activated carriers. So electron carriers, when they are carrying electron, they are almost like the full shuttle bus. So you can think about them as kind of a shuttle bus. So sometimes they are in empty state. So in that case, they're inactive. But then when they end up getting electrons, they become activated. And then they can actually go to another location in the cell and basically donate the electron to, you know, something else. And then you can actually harvest some energy, you know, from that process. So the most common electron carriers in cellular restoration are actually NAD and FAD. So both of them, and NAD actually has a cousin, which is called NADP. So the difference between NAD and NADP is that NAD is involved in cellular restoration, and NADP is involved in photosynthesis. So, you know, they are structurally similar. One has a phosphate, the other one does not. They're also, you know, involved in different process. But the way they get activated is very similar. So, so NAD plus or NADP plus is the inactive form, and then, you know, they can accept electron, and electrons usually actually travel with hydrogen ion or proton, and then basically it gets reduced into NAD plus, NADH or NADPH. And Nigel was asking, can NADP be found in animal cells at all? I'm going to say no, just because it is involved in photosynthesis and our cells do not do photosynthesis. But I'm not 100% sure, so go ahead and look it up and see if that's the case. But I think not, because our cells, you know, have no use for NADP. It's involved in photosynthesis. Okay. And then so the NADH or NADPH can actually donate the electron, you know, later on to something else, and then the cells can harvest the energy from that. Okay, it's not bad. So the question is, which of the following? So remember, we have pairs between NAD plus and NADH, or between FAD or FADH2. Which one is the reduced form? And the correct answer is the one with the H. So the shortcut to memorize it is, remember, electrons usually travel with hydrogen ion. So that's why, whichever form that has hydrogen is the reduced form, and that's the oxidized form. And I think we still have the same people on the leaderboard. Okay, so let's talk about cellular respiration. So it mainly involves 3 different pathways. So it is actually pretty complex. The first pathway is glycolysis. So glycolysis is literally the process that split glucose, which is a 6-carbon molecule, into 2 3-carbon molecules. So the 3-carbon molecules are pyruvates. So one glucose molecule basically goes through, there are 10 steps in glycolysis. It's a linear pathway. And then you end up producing 2 molecules of pyruvates. Without going into too much detail, what should you know about, if you're taking a biochemistry class, you probably have to basically memorize every single step in those 10 steps and what the enzyme is involved, how it's regulated, blah, blah, blah. But, for the Gen Bio class, what you should know is where is it happening in the cell. So glycolysis, regardless of whether it's bacteria or eukarya, it always happens in the cytoplasm. And also how many ATP is generated. And so for that, glycolysis actually generate 2 ATP. And you can actually see that in the first half of glycolysis, you need to put in 2 ATP to get the glucose converted into the favorable kind of conformation. And then in the second half of glycolysis, you end up recovering 4 ATP. So the net gain of ATP through glycolysis is actually 2. And then in the process, you also end up producing 2 NADH. So 2 NAD plus goes in there, and then it gets reduced and give you 2 NADH. And if you break it down, break down the 10 steps of glycolysis, you can divide it into 2 phases. So the first phase is also called the energy investment phase, because you literally have to invest 2 ATP molecule, so that you can actually break this 6-carbon molecule into 2 3-carbon molecules. So you end up producing 2 glyceraldehyde 3-phosphate, or G3P. And then in the second phase, which is also the energy payoff phase, this is where you are harvesting 2 ATP per G3P, but then 4 ATP altogether. And also you can harvest 2 NADH from the payoff stage. So that's glycolysis. Basically know how many ATP you can get from glycolysis, and where it's happening and stuff. The ATP you are that's being produced during glycolysis is the mode is called substrate level phosphorylation. So this is a picture showing actually the last step of glycolysis, and you can see that. So remember, glycolysis, the end product is pyruvate. So this last step, the substrate is phosphoenolpyruvate. The enzyme is pyruvate kinase. And basically, the pyruvate kinase will take a phosphate group from phosphoenolpyruvate and then give it to ADP. And then you end up synthesizing a molecule of ATP, and then the phosphoenolpyruvate gets converted into pyruvate. So substrate level phosphorylation is literally the phosphate group come from one of the substrate, and then the enzyme take it from the substrate and give it to ADP. Okay. So this one is fairly straightforward. Says what's the product of glycolysis, and the correct answer is pyruvate. Glucose is the substrate, and then you don't produce CO2 or water until later in cellular respiration. So glycolysis does not give you those two things. Okay, so that's glycolysis. You end up producing 2 molecules of pyruvate from 1 molecule of glucose. So guys, when we count ATP, you always kind of count it from the perspective of 1 glucose. And then, so after pyruvate is produced, the pyruvate will actually, in eukaryotic cells, will actually go into the cytoplasm, so go into the mitochondria. And then inside the mitochondrial matrix, there is this transition reaction, which is also called pyruvate oxidation. So basically, pyruvate gets converted into a 2-carbon molecule called acetyl-CoA. And then, so one of the carbon is lost in the form of CO2. And then in the process, another NADH is produced. So basically, this pyruvate oxidation, and then of course the coenzyme A gets added to the 2-carbon molecule, so that generate acetyl-CoA. So this one reaction will basically completely oxidize one of the carbon in pyruvate. It gets lost in CO2. And then the other 2 carbon are, being combined with coenzyme A, and then it will also give you 1 NADH in one turn. But remember, glycolysis give you 2 pyruvate. So the net count for, if you're counting what are you gaining from this reaction, is actually 2 NADH, and then of course 2 carbon dioxide. And the acetyl-CoA that's generated from the pyruvate oxidation will basically feed into the next pathway, which is Krebs cycle. So Krebs cycle, the reason it is called a cycle is actually because it's a cyclic pathway. So going in there, we have this 2-carbon molecule called acetyl-CoA, and then acetyl-CoA combined with a 4-carbon molecule called oxaloacetate. And then they form a 6-carbon molecule called citrate. So that's why Krebs cycle is also called TCA cycle or citric acid cycle, because the first product of the first step is citric acid or citrate. Krebs cycle is a cyclic pathway, so it's actually very different from glycolysis. And there are overall 8 steps in the Krebs cycle, and without going into too much detail, once again, the thing you should know about Krebs cycle is, where is it happening? So in eukaryotic cells, it's happening in the mitochondrial matrix. In bacteria, remember bacteria do not have organelle, so it's actually also happening in the cytoplasm. And then what are your gains, right? So what do you gain out of the Krebs cycle? And if you look at one acetyl-CoA molecule feeding into Krebs cycle, so it goes through the cycle, and then basically both of the carbon get completely oxidized and lost in the form of CO2. And then you end up generating 1 ATP or GTP, which is kind of a close cousin of ATP, so people count them as the same. And also 3 molecules of NADH, and then 1 molecule of FADH2. But remember, when we're counting, how many ATP you can gain, we're always counting from the perspective of glucose. So you have to time everything by 2. So the energy gain is actually 2 ATP for Krebs cycle, per glucose molecule, 6 NADH, and then 2 FADH2. Yeah, so Kyle said that Labster had a different yield for the Krebs cycle. I think Labster, I was looking at that table. I think they're counting the 2 ATP from yeah, they kind of had a funny way of counting it. I think they were saying, I think they were saying 8 NADH, And the reason they're saying NADH is because they're counting the pyruvate oxidation step too, I think, yes. So the only thing that's the discrepancy is the NADH. And the reason they're saying that is because they're counting the 2 NADH that's being generated in the pyruvate conversion or pyruvate oxidation step. Although, you know, strictly speaking, that step is not part of Krebs cycle. So, you know, that's where the discrepancy comes in, okay? And so, yeah, so also, so, you know, CO2, same thing. So, you know, 2 of the CO2 come from the pyruvate conversion, and then 4 CO2 actually comes from the Krebs cycle. So yeah, if there is a question asking about the Krebs cycle yield, use the slide. And you know, Krebs, yeah. So yeah, Webster, it is combining, you know, this one step that's outside of the Krebs cycle. Usually, you know, when we ask about the yield and stuff, we're, you know, I mean, look for the language of the question, but if it's very specifically says Krebs cycle, you know, that step above that, you know, shouldn't count as part of the Krebs cycle. Okay, so Krebs cycle, you know, when we're talking about cellular respiration, of course, our main focus is energy. So that's why we're mainly counting APP and how many NADH is being generated, how many FADH2, because, you know, these 2 electron carrier can basically take the energy, take the electron to the electron transport chain, and then, you know, they can also generate more APP from that. But the importance of Krebs cycle is, you know, way beyond, you know, giving you some energy. And I just put up this slide just to show you guys that point. Don't really need you guys to remember the detail. But, you know, basically the Krebs cycle, a lot of the chemicals that are generated in different steps can be funneled outside of the Krebs cycle, and then they can be used for other biosynthesis. So citrate, for example, can actually go out of the Krebs cycle and then get into other pathway to produce fatty acids and also steroids. And then once you have those produced, you know, those things can actually give you membrane material, give you, you know, triglyceride, you know, all kinds of stuff. And then this one over here, alpha keto glutarate can actually be converted into glutamate, and glutamate is actually a neurotransmitter. So there are actually neurons that use glutamate as a neurotransmitter, and glutamate can actually be converted into nucleotides, and then, you know, the cell can actually use nucleotides as a substrate for synthesizing RNA or DNA. And glutamate in other pathways, glutamate can also be converted into amino acids, and then cells can actually use those as building block for synthesizing proteins. So basically, you know, in addition to giving us some energy, the Krebs cycle are actually producing a lot of different chemicals that can be shuffled outside of the pathway and then can be used as, you know, building blocks for synthesizing other things. And so, you know, you produce these NADH and FADH2 from Krebs cycle and also from glycolysis and the pyruvate oxidation. But, you know, the cells actually cannot use NADH as a cellular energy. So it actually really needs to feed into the electron transport chain, or sometimes also called electron transport system, so that you can actually cash in from the NADH and FADH2 and convert them into ATP. So that's mainly the function of electron transport chain or electron transport system, is basically convert the electron that's being carried over here into that's usable energy in the form of ATP. The cellular respiration is also called aerobic respiration because you do need oxygen. So the regular electron transport chain use oxygen as the final electron acceptor. So electrons are basically bouncing from one party to the next, and then from, you know, from the next party to the next party. But eventually the electrons are accepted by oxygen, you know, converts oxygen into water, and you know, and then, you know, we can actually synthesize a whole bunch of ATP in that process. So cellular respiration, you know, animals, a lot of the bacteria use it, and then fungi, protozoa. So, we all perform, and plants actually, we all perform cellular respiration.
And where is water produced in this?
The water is produced in the last enzyme complex over here. Is that what you're asking?
Thank you. Yes,
Yeah.
thank you.
Okay. So this is...
Sorry, just I wanted to ask something about, so the water then is produced in the electron transport chain?
Yes.
Only?
Yes.
Okay, got it. Thank you.
Mm-hmm. So, so actually this graph is a pretty good summarization of everything we have talked about so far. So remember in eukaryotic cells, glucose gets converted into pyruvate in the cytoplasm, and then the pyruvate goes into the mitochondria, into the matrix of mitochondria, and then in the matrix it gets converted into acetyl-CoA, and then the acetyl-CoA feeds into the citric acid cycle or Krebs cycle, and so all the CO2 that are being generated can basically be released. They go out of the mitochondria and go back into the cytosol. The NADH and also the DH2 generated from Krebs cycle will basically feed into the electron transport chain, which is basically the last process of this whole really long chain of process. And the electron transport chain, the basic idea is basic. So, the electron donors like NADH can donate electron to one of these enzyme complex that are embedded in the mitochondria in the membrane. And so the enzyme complex accept the electron and then can actually use the energy to basically pump some of the hydrogen ions through the membrane. And in the last step, we're using oxygen as the final electron acceptor, and that's where oxygen gets converted into water. But yeah, so the basic idea of electron transport chain is actually using the energy from this whole bouncing of electron from one party to the next to pump hydrogen ion across the membrane so that you can establish a very steep hydrogen ion gradient across the membrane, okay? And so in eukaryotes, basically this whole process happens on the inner membrane of mitochondria, and that's because the enzymes you are relying on doing the work are embedded in the membrane, so they cannot happen in the cytosol or in the matrix because these are actually integral membrane proteins. And so NADH actually feeds into the first enzyme complex, and the enzyme complex, use some of the energy to pump hydrogen ion across the membrane. And so over here inside, that's the mitochondrial matrix. And on the other side, remember mitochondria has two layers of membrane. This is the inner membrane. There should be an outer membrane over here. So it's actually accumulating hydrogen ion in this intermembrane space. And then the electron bounce from the first enzyme complex, which is also called NADH dehydrogenase complex, to ubiquinone, which is an electron carrier. Ubiquinone carries the electron to the next complex, which is called the cytochrome C reductase. The reductase can also pump some hydrogen ion across the membrane. And then electron goes to cytochrome C. Cytochrome C carries the electron to the last complex called the cytochrome C oxidase complex. And it's the oxidase complex that convert oxygen into water. So basically oxygen accepts an electron, and remember hydrogen usually travel with electron, and then it gets converted into water. And this complex can also pump some hydrogen ion across the membrane. And this basically end up generating a very steep hydrogen ion concentration gradient. And so I think we already talked about the electron chemical gradient last time. So basically it is a chemical gradient because you have a lot more hydrogen ion on one side than the other. That causes the pH difference because more hydrogen ion means it's more acidic than the other side. It's also an electron gradient. Because hydrogen ion carries positive charge. So that means this side of the membrane is more positively charged than the other side, which is more negatively charged. So this whole electron chemical gradient is also called proton motive force because it's a proton gradient, and cells can actually use this proton gradient to generate basically a lot of ATP. And so the way it works is when these hydrogen ions are flowing back into the matrix of mitochondria, so now it's energetically favorable because you already have a very steep concentration gradient. They flow back into the mitochondria, into the matrix through this enzyme called ATP synthase. So ATP synthase, this part of it is kind of an ion channel that allow the hydrogen ion to flow across. And then the hydrogen ion crossing actually allow the channel to spin. So this is actually a really cool enzyme that can literally, spin around. And then it uses the energy to synthesize ATP. So you're taking ADP and phosphate and then converts them into ATP. So this is the second mode of generating ATP, and it's called oxidative phosphorylation. Substrate phosphorylation, you're taking a phosphate directly from a substrate and then give it to ATP. In oxidative phosphorylation, you're actually using energy generated from proton motive force kind of indirectly to fuel the synthesis of ATP. So that's a huge difference. And in general, if you want to calculate how many ATP you can get from the electron carrier, one NADH can give you 3 ATP, and then one FADH2 can actually give you about 2. So the reason FADH2 give you fewer ATP is because it feeds into electron transport chain in a later step than NADH. So NADH feeds into the first enzyme complex. FADH2 actually feeds into the second enzyme complex. So NADH actually allow more hydrogen ion to be pumped out and then generate a steeper gradient, and then you end up getting more ATP. So that's cellular respiration. Fermentation is another mode for cells to get some energy from things like glucose. And there are different modes of fermentation. So in eukaryotes, so in our body, for example, our muscle cells can actually go through this lactate fermentation and basically produce lactic acid as kind of a side product. The point of fermentation or any kind of fermentation is actually to regenerate NAD+. So if you guys remember from glycolysis, 2 NADH is produced. And if the cell can perform for cellular respiration, eventually that NADH can feed into the electron transport chain, and then you can cash in as some ATP, and then it gets converted back into NAD+. But if there isn't any oxygen available, or if the cells just don't have the machinery to go through electron transport chain, the NADH actually end up being stuck, and it needs more NAD+ so that it can actually extract more energy from glucose. So that's why the cell will actually go through some kind of fermentation to regenerate some NAD+, so that it can actually go back to participate in glycolysis. And fermentation, it's incomplete oxidation of glucose. So, basically pyruvate will, react with something, and then NAD+ gets generated, and you end up producing some product. So lactic acid fermentation, you end up producing lactic acid as the side product. Alcoholic fermentation, you end up producing alcohol as so ethanol as a side product, and also CO2. And so alcoholic fermentation, it's happening in a lot of bacteria and also yeast, and humans, of course, end up taking advantage of it. So we end up using that process for baking, also for generating your favorite beverages or wine or or beer, and stuff like that. And this last slide is actually, it's kind of pretty cool because in animals, it's mainly lactic acid fermentation, but in microbes, so bacteria and also yeast, there are actually all kinds of different fermentation. And if you look at it over here, so these bacteria, Propionibacterium, they actually go through this kind of fermentation and then end up producing propionic acid, and that actually gave us a Swiss cheese. Lactic acid fermentation, so Aspergillus, which is a fungus, Lactobacillus, which is a bacteria, and Streptococcus. We end up getting all kinds of food product from that process. So cheddar, yogurt, your yogurt actually contains a lot of lactobacillus, and also soy sauce. So all come from lactic acid fermentation. Yeast, of course, can produce alcohol. So that gave us, wine and beer. And Clostridium, which is an anaerobic bacteria, they can actually go through fermentation and produce isopropanol or acetone. And that actually gave us things like nail polish remover, because that's something that contains acetone. And then acidic acid fermentation actually gives us vinegar. So you actually see that even though for the microbes, the whole purpose of fermentation is to regenerate some NAD+, it actually give us a lot of food and also other products that we can use on a daily basis. Whoa, okay, so this is a tough one. And we actually have the majority, the one with the highest vote is actually the wrong answer. So 15 people voted for cytoplasm, 10 people voted for mitochondria. So remember, bacteria do not have mitochondria, so that's definitely the wrong answer. And so I'm so glad I put this one in as a question. So this is still showing eukaryote, right? But the point is, all the enzymes that you need for electron transfer chain, they are embedded in the membrane. And so this one actually does show you, so you can see NADH feeds into the first enzyme complex, FADH2 bypass the first one, and it can only feeds into the second enzyme complex. But the whole idea about electron transfer chain is these complex can pump proton across the membrane. So it cannot happen in the cytoplasm. You really do need membrane for that to happen. So bacteria, actually electron transfer chain, all the enzymes are embedded in the cell membrane or plasma membrane. What part of respiration are happening in the cytoplasm? Glycolysis and Krebs cycle. Okay, so that changes things. So we have Charles, Bahar, Roop, Brenda, and Anina on the leaderboard. Okay, so this one is just simple math, and basically to oxidize one glucose into two pyruvate, you have to put in 2 molecules of ATP. You end up generating 4 molecules of ATP, and the net gain is 2. Very good. So between fermentation and cell restoration, the only thing they share is glycolysis, which is actually a very ancient pathway. And let's see, we have Groove on the leaderboard, on the podium, Bahar, and I think it's Charles. Okay. And who else is on the leaderboard? Brenda and Anina are also on the leaderboard. Good job, guys. And so before I sent you guys into a break room for discussion, I just want to show you guys a couple of things. So each lab, I guess there is this learning link, page. And last time, so, remember with the last quiz, people were having trouble with the short answer questions and stuff. So I have added this section on the study guide. So these are things when I used to have exam for the lab, these are the things I listed for this particular lab. So you guys can actually look at the study guide question. It's not a lot, but basically these are the things that you should know about cellular restoration and fermentation, kind of the level of detail. And also, today's the discussion, I added a couple of short answer questions, and reduce some of the lobster questions. When you take them out of context, some of you guys were saying, well, it kind of doesn't make sense. It's been a couple of days since I did the simulation, so I took those out and added a couple of short answer questions. So definitely take advantage of the group setting and talk with your group about the answers. And either time is going to be extended, or I will reduce the number of questions. I haven't completely decided on it. I'll definitely make a decision between tomorrow, and I'll let you guys know in the announcement. And there is a quiz next week. So anything between what's the first week and then between, so cell restoration, cell membrane, and cell structure, right? Because macromolecule was already covered in the last one. Well, it's not cumulative. It's cumulative means it's everything, right? So it's everything that's not being covered from the first quiz that we have done since. So 3 labs, I think. Okay. And just the lab report, I posted a couple of examples, so highly recommend you guys look at those examples just to give you a sense of, those are very different labs because it's for a different class, but just give you a sense of how other students structure their lab report. And I'm not confident that I will be able to have enough time to give you feedback, so we're not going to do a draft submission. We're just going to stick with the same deadline. Okay. All right, so I'm gonna start creating the breakout room, and Kate, I guess you can stop recording.